Assalamu alaikum students, welcome to this lecture on Watson and Crick model of DNA. In 1951, Erwin Chargaff discovered specific ratios of nitrogenous bases in DNA. He observed that the ratio of adenine is always equal to thymine, means the amount of adenine is always equal to amount of thymine, and the amount of guanine is always equal to amount of cytosine. These are actually the nitrogenous bases, and Erwin Chargaff discovered the ratios of these nitrogenous bases in any DNA. And he observed that purines and pyrimidines are in ratio 1:1, means the purine, which include adenine and guanine, are in ratio 1, and thymine and cytosine, which are also in 1. Means purine and pyrimidine both are in ratio 1:1, and means purines are always equal to the amount of pyrimidines in DNA. And this principle, which he called Chargaff rule, which states that the amount of purines are equal to the amount of pyrimidines in DNA. Next on we have other scientists, Maurice, Wilkins and Rosalind, and these two scientists worked on the structure of DNA by X-ray diffraction analysis. They discovered that the width of the DNA is 2 nanometer and the length of the DNA is 3.4 nanometer. Each of this strand is 3.4 nanometer, and the distance between the nucleotides, this nucleotide and this nucleotide, is 0.34 nanometer. And this is the structure of messenger RNA, or you can simply say RNA. And in RNA, the distance between nucleotides is 0.7 nanometer. And these two scientists worked on structure of DNA, and they said that DNA is not a single stranded, instead the DNA is double helix loop, which wind around each other and creates a loop-like structure. Next on Watson and Crick model, which was proposed in 1953 by Watson and Crick, and they told us that DNA has two nucleotide chains. Like this is the structure of DNA, simple structure of DNA, which is not wounded, just a simple, this structure is like simple structure of DNA. These are the two chains of DNA, two nucleotide chains, and these are the nucleotides. Adenine is bonding with thymine, cytosine is bonding with guanine, guanine with cytosine, thymine with adenine. And these two chains are anti-parallel, means if this chain is starting from 5' end and going towards 3', the other chain is starting from 3' to 5', means they are anti-parallel, means they are opposite to each other. And how can you differentiate the 3' end and 5' end? In 5' end, phosphate group is on the above. Here you can see this is the 5' end, phosphate group is on the above, and this is the 5' end and phosphate group is here. And in 3' end there is no phosphate on the corner. Here is also no phosphate, it means this is 3' end. And he told us that chains are anti-parallel. One runs from 5 to 3 and other 3 to 5. I already told you. And the base pairing, adenine bonds with thymine and guanine with cytosine. Okay, so we have next DNA structure and base pairing. We have already discussed about it, that adenine pairs with thymine and guanine pairs with cytosine. And here you can see this is adenine and this is thymine. Adenine bonds with thymine, and there are two hydrogen bonds between adenine and thymine. And this is cytosine and this is guanine. And guanine and cytosine both have 3 hydrogen bonds in between them. And these hydrogen bonds are very weak and they can be easily broken down. Next, third point shows that base pairs per turn are 10. Okay, so this is one loop. Okay, so this has 10 base pairs, 10 bases, 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. These are now not counted because these are in the middle of this loop. So here are 10 you can say like nucleotides in one loop, 10 bases. And the complementary strands, the sequence of nucleotides varies among species and individuals. This is one strand, and this strand is complementary to this strand, means if there is adenine, there will be thymine. If this is cytosine, there will be guanine, means they are complementary to each other. And they varies among different species and individuals. Mean if in one individual, here first nucleotide is adenine, it might be different in another individual which has like thymine in thymine is the first nucleotide in their gene pair. So this was all about this topic, Watson and Crick model of DNA. If you have any questions related to this topic, you can ask in the comments. Thank you so much.